عليكم السلام والرحمة. السلام عليكم. Good afternoon, everybody. Type. So today we're gonna discuss the nitrogen removal and urea cycle. But before that, we can have a quick revision on the electron transport chain. As we mentioned, guys, stop talking. As we mentioned before, to understand electron transport chain, you need to understand the structure of mitochondria. And we mentioned that mitochondria have outer membrane, inner membrane, and between them it have intermembrane space, and the inner membrane form a cristae. There is a protrusions from the inner membrane that form the what they call it cristae, and we have the mitochondrial matrix. Inside the mitochondrial matrix, citric acid cycle take place, fatty acid oxidation take place, and those metabolic pathway are the major pathway for energy production. They produce NADH, so they have to be very close to the electron transport chain. So they give the NADH directly to the electron transport chain, which is located within the inner mitochondrial membrane. One of the main features of the mitochondrial membrane that they are impermeable to ions, impermeable to any other molecules. So for any molecules, even for ions, small ions, to get inside the mitochondria or to get outside the mitochondria, they need transport. Without transport, they cannot get in or outside the mitochondria. The electron transport chain, as we mentioned, have 5 complexes. Complex 1 basically accept the electron from NADH. 2 FMN, which is take the hydrogen electron and is converted to FMNH2. Complex 2 is basically taking the fat H from the succinate dehydrogenase, which is the only reaction in the citric acid cycle that produce fat H. So both complex 1 and 2, they give their electrons with the proton hydrogen to coenzyme Q, which take the electrons, accept, and donate them to complex 3. Complex 3, same, they take the electron from cytochrome C and provide it to complex 4. Both in complex 3 and 4, there is heme containing protein. That means they have ferrous. So they get just reduced, they accept the hydrogen and electron. So from Fe plus 3, they become Fe plus 2 because they accept 1 electron, so they became less positive. In complex 4, the electron is with the proton interact with oxygen and give rise to water. So this series of reaction, they just show that there is electron acceptor and electron donor. It starts from NADH and fat H. They are the main electron donors. Both of them, NADH to complex 1 and fat H2 to complex 2, and both of them give the electron to complex 3 and then complex 4, and at the end this will produce water in the presence of oxygen. And that's why when we always keep talking about aerobic metabolism, because if there is no oxygen, this series of reaction will stop, because the final acceptor of the electron is oxygen. So if you don't have oxygen, the electron transport chain will not work, because it needs at complex 4 to interact with oxygen and produce water. And that's why this process of producing energy from NADH is oxygen dependent. No oxygen, you cannot use NADH or fat H to produce energy. And that's why we said that citric acid cycle is just aerobic, because if there is no oxygen, all fat and NAD will be saturated and they will become fat H and NADH. If they cannot be consumed to produce energy in electron transport chain, the whole reaction will stop. So from complex 1 to complex 4, we have a flow of electron from donor to acceptor, donor, acceptor, until it gives water. This flow of electron through these complexes create energy. And the energy that is created by the flow of electron is used to pump the hydrogen from inside the mitochondria to the intermitochondrial membrane. So this bump of hydrogen through complex 1, 3, and 4 will create gradient. So in the intermembrane, the hydrogen will increase. So there will be pH gradient. The hydrogen concentration in the membrane, in the intermembrane, will be higher than the hydrogen concentration in the matrix. At the same time, it is positive charge, the proton. So there will be more positive charge in the intermembrane than in the matrix. This will create a force that will force this proton to try to get back to the matrix. And the only way for those protons to get back to the matrix is through the ATP synthase, complex 5. So it will push back to the matrix by or through complex 5 ATP synthase, and the energy or conformational change happen while this proton is bumped back is used to phosphorylate ADP into ATP. And that's why we call it oxidative phosphorylation. Oxidative because the flow of electron at the end will meet oxygen to produce water. So it is oxidative process. And oxidative phosphorylation because the end product is the phosphorylation of ADP to ATP. So the function of electron transport chain, by other words, is termed as oxidative phosphorylation process. Okay, so then we have the flow of electron and we have the production of ATP. Then the question, why not H produce 3 ATP while fat H produce only 2 ATP? Yes. So for fat H2, it bypass complex 1 because the first bump is through complex 1. But for fat H2, it pass complex 1 and it starts complex 3. That's why it will be only bumping only 2 hydrogen and will result in only 2 ATP. While for NADH, it is the pump of through complex 1, 3, and 4, so it is 3 ATP. Then you have to understand that there is some chemicals or some molecules, they have the ability to inhibit or to suppress a specific component of electron transport chain. And we gave one example, which is example of electron transport chain inhibitor.
Is it like used for poison?
Yes, for poison, but cyanide, yes, but I gave example. Cyanide I didn't mention in the PowerPoint, but in the PowerPoint I give a specific example. It is oligomycin. Okay, oligomycin. Then we talk about the uncoupler. There is a protein that is because we said from the beginning that the membrane of the mitochondria are impermeable. Some proteins can make holes in the membrane. So then the mitochondrial membrane will become permeable. This will affect the process of production of ATP because when the hydrogen is pumped into the intermembrane, then it can come back to the matrix through other pathways, not necessarily through ATP synthase. So this will uncouple the oxidation with the phosphorylation because there will still be oxidation as electron will move through the complex until bind to oxygen and give water. But there will not be simultaneous phosphorylation because the pump of hydrogen can get back through this hole or through this transporter. And those uncoupler, as we said, they disseminate the heat. They waste the energy. So instead of producing ATP, the energy is converted into the energy produced with this pump will be converted into heat. Okay? And we gave example also. In the newborn, yes, they have what we call it brown adipose tissue. Yes, brown adipose tissue. Okay, that's it for the electron transport chain. Today we will talk about different topic. It is the nitrogen removal and urea cycle. Okay, so in the body can store fats and can store carbohydrates, but the body cannot store amino acids. So these amino acids, either if you eat a protein, so this protein either immediately used to synthesize new proteins, or it should be catabolized, it should be degraded. But the body cannot store the amino acid as glucose, for example, because the body have the ability to store the glucose in form of glycogen. The body have the ability to store fats in the adipose tissue, but not for the protein. Okay? So it is necessary for the body to always anabolize and catabolize amino acid. Okay, so what is the problem then of continuously catabolizing amino acid? The problem is in this NH3, the ammonia. Any amino acid have carboxylic group and have amino group. Any amino acid. These are the rest for the amino acid. The removal of amino group is mandatory. If you want to catabolize this amino acid, you have to remove the amino group. So it is mandatory to remove the amino group to catabolize the amino acid, because when you remove this amino group, you will get just like a carbon skeleton of a keto acid. This can be used in different pathways. For example, if you have amino acids and you give you take from the amino acid this amino group, it will become alpha ketoglutarate. If it was glutamate, for example, glutamate, if you take amino group, it becomes alpha ketoglutarate. Then it can go into the citric acid cycle. But the problem will be in the amino group. And what is the problem of amino group? The problem that the free ammonia is toxic. So if you just cut this, for example, glutamate, if you just cut the amino group and convert it to alpha ketoglutarate, this you can use it in other pathway, but the problem will be in this ammonia because it is toxic for the body. So it need to be either converted into non-toxic components or transferred to other keto acids, but cannot be left in the circulation. So either, as I said, either incorporated into other compounds or excreted out as urea. So the urea is a non-toxic compound that can carry the ammonia and can be excreted in the urine without any complication. So there is two types of reaction for the removal of the nitrogen group or for removal of ammonia. There is a transamination reaction or oxidative deamination. Transamination is simply you just move this amino group from one compound to other compound. So it is just exchange. You are just incorporating the amino group into other molecule. The oxidative deamination, you cut and remove the ammonia. So the transamination, the oxidative deamination give ammonia, and transamination can give some amino acid like aspartate. And both aspartate and free ammonia are used to produce urea, which, as I said, it is the non-toxic form of the ammonia. So whatever amino acid we eat, if we don't use it for synthesis of new amino acid, the amino group either incorporated into other molecule or used to form urea. So the urea cycle, the cycle that is responsible of converting this toxic ammonia into non-toxic urea, it is the main disposal of nitrogen in the body. And if this cycle is for any reason is not working, then the consequences are very dangerous, as we will discuss at the end of the lecture, because this will lead to accumulation of ammonia, which is toxic for the organs. So, as I said, two types of reaction, either transamination reaction, and here the amino group is transferred from first step catabolism involves transfer of amino group from amino acid to keto acid. For example, from alanine to alpha ketoglutarate or to pyruvate. Alpha ketoglutarate is the main acceptor of amino group from most amino acids. So the acceptor of the amino group from most of the amino acid, because we will have a lot of amino acid, we will have a lot of amino group. So the main acceptor is alpha ketoglutarate because it is abundant from through citric acid cycle. And when alpha ketoglutarate receive amino group, is converted to glutamate. Glutamate is amino acid because if you remove the amino acid, the amino group from amino acid, it will become keto acid. If you add amino group, it become back amino acid. This transfer of amino group from one carbon skeleton to another is catalyzed by family of enzyme called aminotransferase. Simply, they transfer ammonia from one molecule to other molecule. That's why they call them, or they transfer the amino group from one molecule to other. That's why they call aminotransferase. They transfer amino group. And those aminotransferase are found both in cytosol and mitochondria. So again, other example of metabolic reactions is here, the transamination, like gluconeogenesis, both in mitochondria and cytosol. And here also transaminase or transamination reaction are also found both in cytosol and in mitochondria. Each aminotransferase have a specific name, and they named after a specific amino group. It's the same as in the citric acid cycle and in glycolysis. Usually, glucose 6-phosphatase mean that it work on glucose 6-phosphate. It remove the phosphate group. Here it is the same. If, for example, alanine aminotransferase, it simply means that it work on amino acid alanine, and it remove the amino group from alanine to give it to other, transfer it to other keto acid. So here ALT, alanine aminotransferase is called ALT. So it work on alanine. It just give the amino group to alpha ketoglutarate. Alanine without amino group will become pyruvate, and alpha ketoglutarate plus the amino group will become glutamate. So it is just simple transamination reaction. Aspartate aminotransferase, those are the most popular. AST, it work on aspartate, and it convert, it gave the alpha ketoglutarate the amino group. So alpha ketoglutarate will become glutamate, and aspartate will become oxaloacetate. And AST and ALT, they are the most abundant aminotransferase. They are very popular, and that's why they have a clinical importance. Have you ever heard about liver function test? Liver function test. Yeah, for hepatitis, if we suspect the liver is damaged or affected by or inflamed or whatever, so we ask for liver function test. It is a series of tests show if the liver is functioning normally or not. If you ever looked into the liver function test, you will find that they measure the ALT level and the AST level. Okay? They measure these aminotransferase, alanine aminotransferase and aspartate aminotransferase. Why they measure them? Because they are very abundant in the cytosol. If the cells are destroyed, the liver cell will release a lot of ALT and a lot of AST. So in the circulation, the patient with the damage of the liver cell will have high level of ALT and high level of AST. So this investigation or this test show that the liver, there is a problem in the liver, because the level of AST and ALT, they should be inside the mitochondria and cytosol of the cell. So if it is damaged, it will release into the blood and it will show. So this is just an example of how people make use of the biochemistry knowledge in clinical application. So they are the two most important, ALT and AST, and as we said, they are mainly in the liver because the urea cycle is mainly in the liver. So how transamination reaction happen? Or any transaminase, any enzyme, transaminase, it needs coenzyme called pyridoxal phosphate. It is vitamin B6. It is the active form of vitamin B6. Okay? So for any transamination reaction or for any aminotransferase enzyme, this coenzyme is needed. It is vitamin B6. And it acts as a carrier of the amino group. We said that aminotransferase, they transfer amino group from one molecule to other molecule. This transfer is happening through this pyridoxal phosphate. So here, for example, here you have glutamate. It is amino acid, and it have amino group. So what will happen to this? It gives the pyridoxal phosphate takes this amino group from glutamate. So glutamate will be converted into alpha ketoglutarate. So glutamate minus amino group is alpha ketoglutarate. And then the pyridoxal phosphate will bind to the NH3. Pyridoxal phosphate then will work on other keto acid, oxaloacetate. It will give the amino group to oxaloacetate, and oxaloacetate will be converted into aspartate and free pyridoxal phosphate. Pyridoxal phosphate can interact again with amino acid and can carry again amino group and can give it to other keto acid, which accept the amino group and convert it into amino acid. So this is a how amino transferase reaction happen. The other type of reaction, because we said there is two reactions to remove the amino group, either transaminase or oxidative deamination. So the oxidative deamination of amino acid, here there is no acceptor. Here the amino group is from here we have glutamate, the same, but the difference here with the glutamate dehydrogenase, you can directly take the ammonia from the glutamate without adding it to another keto acid, and then you will get alpha ketoglutarate. So this direct deamination, you just take an amino group and convert this amino acid into keto acid. And the opposite also can happen. You can directly add the amino group to keto acid, alpha ketoglutarate, and convert it to glutamate. And the reaction is catalyzed by enzyme called glutamate dehydrogenase. And this reaction can produce NADH also. So if you convert amino acids into keto acid with this reaction, you can produce NADH. So you can produce energy from deamination of amino acid. And that's why in the first slide we said that to catabolize the amino acid or to use them to produce energy, you need to remove the amino group. Removing amino group by deamination can produce NADH. So each amino acid like this can give one NADH, like 3 ATP. And this reaction take place mainly in liver and kidney. So this deamination reaction, they mainly happen in the liver and in kidney. They provide keto acids that can enter central pathway. So by deamination, you get alpha ketoglutarate. Alpha ketoglutarate can directly get inside the citric acid cycle. How the ammonia is transported in the blood? We said that the ammonia is toxic. So how the body can transport the ammonia from peripheral tissue to the liver? Because in the liver is the main organ that can convert ammonia into urea. The organ that can convert the toxic ammonia into non-toxic urea. So what about amino acid in the muscle? What about amino acid in different tissue, in adipose tissue? How we can transport them to the liver? There is two ways of transporting ammonia without intoxicating the body. So the first is the use of glutamine synthetase. So what this enzyme is doing, it combines ammonia to glutamate, and then it produces other amino acid called glutamine. This amino acid can go through the blood to the liver. The other way is to involve formation of alanine by transamination of pyruvate. We know pyruvate is everywhere. From glycolysis, we get pyruvate. So we can use this pyruvate as a carrier of the ammonia. So when we add with transaminase reaction or transamination reaction, when we add amino group to pyruvate, it's converted into alanine. Alanine can be transported through the blood to the liver. And in the liver, this amino group will be converted to urea. So as you can see here, most of the tissue, they gave the amino group to the glutamate. So the ammonia is incorporated into glutamine, and this can go to the liver. From the muscles, the glucose through glycolysis give pyruvate. Through transaminase reaction, this can be converted into alanine. Alanine can be transported back to the liver. In the liver, this ammonia can be, we can get the ammonia by transamination reaction, so we can convert it back to pyruvate, and we can give this amino group to glutamate. And then the glutamate will contribute in production of urea. So we can collect all the ammonia from peripheral tissue to the liver, and we convert them in the liver into urea, send it in the blood, and it will be excreted with urine as a non-toxic compound. So what happened exactly in the urea cycle? In urea cycle, as we mentioned, it is a major disposal form of amino group. So all the amino group in all different parts of the body, they are converted into urea. So this is a major disposal pathway for the amino group. So out of 2 nitrogen of urea molecule, one is coming from free ammonia, deamination reaction, free ammonia, and the other came from aspartate. So each urea molecule have 2 amino group. One of them is coming from aspartate, the other one is just come as free ammonia. As I said, urea is produced by the liver and then is transported into the blood to the kidney to be excreted in urine as a non-toxic. The most important thing here that the first 2 reactions occur in the mitochondrial matrix, and the rest of the cycle occur in the cytosol. So urea cycle like gluconeogenesis. It takes place both in mitochondria and in the cytosol. The 2 reactions of urea cycle occur in the mitochondria. The rest of the reaction take place in cytosol. So what's happening that, as we said, aspartate is the main carrier of amino group. And as we mentioned before, this aspartate is coming from transamination reaction with glutamate. So the amino group from glutamate bound to oxaloacetate, and it gives aspartate and alpha ketoglutarate. Okay? And as we mentioned before, glutamate is coming either from transamination reaction with alpha ketoglutarate and N amino acid, or oxidative deamination reaction from directly removing the amino group from the glutamate. So what happened here, that aspartate, the first reaction in urea cycle, that aspartate bind to molecule called citrulline, and then they gave argininosuccinate, and then fumarate arginine and urea. So you need carbon dioxide, you need amino group. So the amino group, the free amino group is coming only from deamination. So you can see here you have free amino group here. When it bound with carbon dioxide, they produce molecule called carbamoyl phosphate. This carbamoyl phosphate bind to other molecule called ornithine. The combination of ornithine and carbamoyl phosphate give rise to citrulline. Citrulline will bound with aspartate to give aminosuccinate. Aminosuccinate will be hydrolyzed into arginine and fumarate. And then the arginine, through urea synthesis, it will give urea plus ornithine, and ornithine will continue. So it is like as a cycle. At the end of the reaction, you regenerate ornithine. In citric acid cycle, at the end of the reaction, we were regenerating oxaloacetate. So it's the same here. Here you regenerate ornithine. The ornithine will bound with carbon dioxide and amino group to give citrulline. Citrulline will bind with aspartate, and then until it gives urea. The most important thing about this reaction, first it takes place in the liver mainly. In the cell, it takes place both in mitochondria and in the cytosol. Each urea cycle have two amino group. Each urea cycle have two amino group. One of them is a free amino group produced by direct deamination of glutamate. The second one is coming from aspartate. So the amino group here with the direct deamination combine with carbon dioxide to produce this carbamyl phosphate, which interact with ornithine to give citrulline. And here the aspartate bind with citrulline to give argininosuccinate, and the cycle continue and give rise to urea plus regenerating ornithine again. So the first reaction is amino group with carbon dioxide, the carbamyl phosphate, which is bound to or interact with ornithine to give citrulline. So urea cycle is one of the most important metabolic pathway in the body because it is a major disposal of nitrogen. We said from the beginning of the lecture that the body cannot store amino acid. So either use it for synthesis of protein or catabolize it. The problem with catabolizing amino acids is that you will get ammonia. What is the problem with ammonia? Ammonia is toxic. That means the body should handle it carefully. So how we get rid of ammonia? We convert it into urea. Where this process takes place? In the liver. What about ammonia that is occurring in peripheral tissue? It should be transported either in form of glutamine or alanine. Urea cycle takes place inside the liver, both in mitochondria and in the cytosol. Each molecule of urea have two amino group, one free, the other one with aspartate. That's why they are in purple. Urea, aspartate, and amino group. The free one only can come with oxidative deamination of glutamate to release free ammonia. The one of the aspartate is with normal transamination reaction. The first reaction, ammonia, carbon dioxide, carbamyl phosphate with ornithine, citrulline. Citrulline with aspartate, argininosuccinate, and the rest. So overall, you need one molecule of aspartate, one molecule of ammonia, free ammonia, bicarbonate, and 3 ATP, water to get urea. Okay? 2 ADP plus 1 AMP plus, so it is 4 high energy phosphate bonds are consumed in synthesis of one molecule of urea. So it is expensive process for the cell to convert ammonia into urea, but it is very important because if ammonia accumulates, it will intoxicate the body. For each molecule of ammonia, we need 3 ATP. To produce one molecule of urea, we need 3 ATP. We need molecule of aspartate, and we need free ammonia. So this is the hyperammonemia, the accumulation of ammonia in the blood. So the urea cycle in the liver, it has a capacity. So it cannot like just convert whatever amino group is coming. And it keeps the level of ammonia very low. So the level of ammonia in normal people like you, it should be 5 to 35. If it increases from this level, then we call it hyperammonia, and it will be medical emergency because it is dangerous for the body. So when this hyperammonia happen, since urea cycle take place in the liver, so it happen when there is a liver failure. When liver is failing due to infection like hepatitis or due to liver failure for other causes, intoxication with CCl4 or intoxication with paracetamol overdose or whatever, if the liver fail, the liver will not be able to convert ammonia into urea. What will happen? The ammonia will increase in the circulation. Guys, this is the last slide. إذا عايزين تتكلموا شوية أنا ممكن أقعد أنتظر، أنا ما ورايا حاجة، عندي لسه 20 دقيقة. If you want to تموا كلامكم وبعدك نواصل. ناخذ 10 دقائق استراحة إذا ما مستعجلين. ما يعني نتكلم أنا، يعني تتكلموا أنتوا. وأنا ما عندي مشكلة، يتكلموا أنتوا. We'll take 10 minutes. حيتكلموا أنا ولا أنتوا؟ أيوة، لأنه ده آخر السلايد. أنا حكون مستمتع أقعدكم 10 دقائق، لأنه أنا أصلا قاعد أنا المحاضرة الجاية الساعة 2 مع الجروب اللي بعده، يعني أنا قاعد أصلا. أنتوا اللي مستعجلين ولا ما مستعجلين؟ خلاص أتكلم أنا؟ والله لو واحد يتكلم أنا حقعد هنا لحد لما يفضل 5 دقائق وما حخليه يقدر يطلع. اوكي، قلنا انه if the liver fail، السلايد دي هي اهم سلايد في المحاضرة كلها. if the liver fail what will happen? The liver will not be able to convert the ammonia into urea. قلنا الامونيا it's toxic. The normal level of ammonia should be between 5 to 35 micromole per liter. If the liver failed due to hepatitis, التهاب الكبد الوبائي, or due to intoxication or due to paracetamol overdose or whatever, what will happen? The ammonia will level in the blood will increase. It can also be due to genetic defect of the urea cycle or any liver disease. So the blood level of ammonia will lead to hyperammonemia, which is a serious condition. It is a medical emergency because it have direct neurotoxic effect. It affects the CNS, so it can lead to convulsion, coma, and death at the end. It lead to brain edema, so a lot of serious complication. So what is the symptoms of the hyperammonemia? How you know that these patients have a problem or have high level of ammonia in the blood? They have tremor, irtiash, slurring of speech, they cannot speak normally, drowsiness, they get confused, vomiting, cerebral edema, as I said, increased cranial pressure and headache, blurring of vision, at the end, coma, and it can even lead to death. So liver failure, the most dangerous thing about patient with liver failure is the impairment of urea cycle, which lead to hyperammonia, direct effect on CNS, and it can lead to deep coma and death. يعني البيشنت اللي عندهم liver failure they don't die due to liver problems, they die due to high level of ammonia in the blood and CNS complication. Okay? If you don't have question, that's it. عليكم السلام والرحمة.
الحين هني the first reaction occur in the multiple يعني تتكلم عن في first action occur in the liver.
ايوه دقيقة بس. عليكم السلام والرحمة. شكرا جزيلا. شو هذه؟ وردة؟ نعناع؟
لا
شكرا.